The regulatories are favoring process intensification and continuous processes. We have the ICH Q13 and so on. But what I'm hearing from QA and regulatory is basically what they are requiring from a batch process as well. They want to have traceability, a solid definition of what a batch is. They want to focus on patient safety, which means virus clearance in the low pH step, in the nanofiltration, in the chrome steps, controlling bioburden, and then finally a control of product quality with adequate process characterization studies. Basically, I don't see anything revolutionary there in the demands. It's basically what you are doing in traditional batch manufacturing.
Is continuous manufacturing just hype, or can it actually deliver real value for your bioprocess? Today, we are separating fact from fiction and exploring the economic realities of next-generation manufacturing. Welcome back to part 2 of the Smart Biotech Scientist podcast. I'm David Brühlmann, your host, continuing our conversation with Andreas Castan from Cytiva. We will dive into implementation strategies, sustainability impacts, and the decision frameworks every bioprocess engineer and scientist needs when evaluating fed batch versus continuous processing. Let's get started. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech.
I'd like to dive into a bit more continuous part, which is part of the process intensification. You mentioned that you could use that for your seed train to get to higher cell densities, or you could even take it a step further and do your entire production bioreactor run in continuous, or even the downstream or parts of the downstream in continuous. Can you tell us what are different companies doing, what are different models, and why should someone who is now only working in fed batch consider moving into a continuous process?
The advantages with continuous processes, the productivity, for example, in a high cell density perfusion is typically 3 to 5x compared to a fed batch bioreactor. And that means that you can produce the same amount of material in a perfusion bioreactor that is 20-30% the size of a fed batch bioreactor. And then you can build a smaller plant and decrease your capital cost and reduce the HVAC, and that can decrease your operating cost and carbon footprint and so on. But of course, it comes with a cost as well. You increase the complexity. You need to set up a downstream operation that basically works also continuously all the time. You need to prepare large amounts of money, of medium. And you also need to create an overarching automation control strategy to connect the upstream and downstream processes. It adds a little bit on the complexity, and there are a few companies that basically driven by their pipeline needs are moving into continuous, like AstraZeneca and Sanofi and Merck, manufacturing of the future. There are companies kind of looking more into a hybrid approach, having perfusion, multi-column chromatography, and then batch. Enzene, for example, Merck Serono, and then there are a large number of companies which utilize this high seed fed batch N-1 perfusion for example, Lonza, Takeda, Alexion, and they have increased, like I said, their titers by these intensification between 50 and 150%, which they reported the Cell Culture Engineering Conference earlier this year. So it's a diverse toolbox, and you need to understand what your pipeline requires, what your capabilities are, what your bottlenecks are, and how you set up the kind of long-range supply strategy. Whether it works to go into continuous or whether you are better off with an intensified fed batch.
So it looks also like it's a longer term strategic choice, because I would say if you only plan a few batches for clinical production because you're in phase 2, it probably will not make sense to invest that much to get your head around all the complexity in these tools. It's much more viable and beneficial as you're moving into phase 3 and then commercial manufacturing.
Yeah, I fully agree with that. And I always encourage companies I'm working with thinking of keep it as simple as possible. Really, like I've said now a couple of times, understand your product, your material needs, your manufacturing network, your capabilities in the organization, and make a choice based on that and keep it as simple as possible.
Yeah, that's very good advice, to keep it as simple as possible.
But still focus on robustness from the start so that you have a sustainable solution that you're working with.
And that's also where continuous processing is a plus, because if you do it well with the right tools in place, the right control strategies, you have a much higher likelihood to make it robust because of the nature of a continuous process.
Yes, absolutely. Fully agree with that.
Now, there is one thing we should talk about when we talk about continuous processing, because if you run your production bioreactor in a continuous mode, your media consumption will be much higher. And we know media is not cheap. How does the cost of goods equation change as you're moving from fed batch to a continuous process? I assume that cost of media, the percentage will be much higher in a continuous process than in a fed batch process, right?
That's true. When looking in process economy, it really depends on the scenario, basically in terms of how many kilos per year you want to produce. That should be the kind of starting point for an economic modeling. And generally, continuous process requires lower capital costs because the bioreactor is smaller, the plant can be smaller, but higher costs on cell culture media and labor cost also because you need to match your continuous bioreactor with a continuous downstream. However, we've been modeling process economic using Biosolve in order to have an accepted baseline. And for example, looking at a production or productivity of 100 kilos per year, cost of a single-use fed batch compared to a continuous process are very close to each other. It basically levels out each other so that plus minus 10%. We have also looked into, just as you pointed out, the cell culture media cost, and looked into the case of kind of proprietary cheap cell culture media, maybe with a cost of $9 per liter. Then around 20% of the COGS are related to the cell culture media. If you buy a kind of the shelf commercially available medium with a cost of, let's say, $19 per liter, the percentage of the COGS related to the cell culture media are 25%. So it has an impact, but it's not radically changing their business case. But definitely 20% is a big chunk of total COGS.
What were some key learnings you made as you were working with various continuous processes and also as you were working with your clients? What were some maybe of the hard learnings, or maybe you had some positive surprises as well as you were working on these projects?
Yeah, I mean, when working with customers, aha experiences come actually from process economics and understanding the impact of different factors. For example, when scaling out, intensifying by buying more bioreactors and utilizing the downstream line more efficient has a very significant impact. But also scaling up with their model 2K up to 12K fed batch bioreactors and compared that with 500 liter to 2K perfusion bioreactors, we are seeing a tremendous impact on the cost of goods. However, the difference between fed batch and continuous are very minimal in these scenarios. And that is the reason why it's still attractive to utilize these big stainless steel bioreactors of 12K, 20K that are available in the market, because scaling up has such a tremendous impact on the cost of goods, both for fed batch and for a continuous.
Before we continue, biotech founders, I want to talk directly to you. Do you know that feeling when an investor asks about your manufacturing strategy and you freeze? One wrong CMC decision can cost you $2 million and 18 plus months of delays. Yet many CEOs are flying blind on these critical decisions. That's why I created the 1-week CMC strategy assessment. In 1 week, you get a personalized assessment of your molecule and development stage. We identify critical gaps before they become million-dollar mistakes, plus a strategic roadmap for high-stakes investor meetings. One CEO told me, David took me from feeling powerless about our CMC roadmap to confidently pitching investors. An absolute game changer for time-pressed CEOs. Whether you are preparing for Series A, approaching IND filing, or facing scale-up challenges, this gives you confidence to ace investor and board conversations instead of surviving them. Ready to stop second-guessing your CMC strategy? Check the link in the show notes and secure your spot. Because your breakthrough therapy deserves a solid CMC strategy. Now, let's get back to our episode.
Process.
Let's talk about the sustainability part as well, because that's an important one. Do next generation bioprocesses actually deliver environmental benefits? And if how?
Sustainability is a topic that is really engaging our customers community as a whole, and our customers have targets where they want us as Cytiva to contribute to reach these. And we are very involved in the Nimble framework, where we investigate different process templates with respect to sustainability in a way where basically the industry is working together. So we have multiple stakeholders there and align on a way of calculating. And this is a work in progress. So far, there's actually no clear winner comparing fat batch with continuous processes. The carbon footprint for continuous processes is smaller, maybe about 80% compared to a fat batch process. I personally had expected almost an order of magnitude difference, but that wasn't coming out of these calculations. And when looking into the details, the benefit comes mainly from the difference in the HVAC. We mentioned the smaller footprint of the classified areas, smaller clean room area in the continuous scenario. So a bit surprising, a little bit disappointing for me as well, having worked with perfusion for almost 20 years, but that is where we are right now. But as I mentioned, it's work in progress.
Yeah, that's definitely surprising that it's not so different. I would have expected that there would be a bigger difference between the two. I would just briefly touch upon the regulatory side, because the health authorities made a clear stance that they are in favor of continuous processing. So both the FDA and the MEA, they had clearly stated that they are encouraging companies to move forward. And yet certain companies are still on the fence. And I think it mainly comes from regulatory affairs departments or from quality assurance. What do you think? Does the industry need to change, or is there a mentality shift? What is still causing that a lot of people are a bit waiting and seeing before taking the leap of faith?
I think the decision on technology is mainly a business decision driven by the pipeline. We mentioned the different companies which have different pipelines and are therefore taking different decisions. But as you said, I agree with that the regulatories are favoring process intensification and continuous processes. We have the ICH Q13 and so on. But what I'm hearing from QA and regulatory is basically what they are requiring from a batch process as well. They want to have traceability, a solid definition of what a batch is. They want to focus on patient safety, which means virus clearance in the low pH step, in the nanofiltration, in the Chrome steps, controlling bioburden, and then finally a control of product quality with adequate process characterization studies. Basically, I don't see anything revolutionary there in the demands. It's basically what you are doing in a traditional batch manufacturing. Again, I think it's more a decision on the business case that is driving in one or the other direction.
What do you think are the most common mistakes you see bioprocess scientists and engineers make when trying to implement these advanced manufacturing concepts?
I think that's often the scientist or the engineer falling in love with a certain technology and getting very attached to it and pushing for it without looking and understanding actually the situation it is. So I think that is something I'm seeing very much. It underpins the need to understand your scenario and choose the technology after that. Consider robustness already for the first in human. And again, don't go too fancy. Leverage the existing knowledge from your suppliers, from the literature, from conferences, from podcasts like this to be well informed and not blindly move into one technology.
Before we wrap up, Andreas, what burning question haven't I asked that you're eager to share with our biotech community?
It's maybe about the long-term vision. In the beginning, we talked about what I'm seeing to happen in 3 to 5 years. Shall I answer that?
Of course, go ahead.
I have to share that vision. What is keeping really me awake at night is the accessibility of drugs, the high cost that we are seeing. The Gates Foundation challenged the industry by reaching $10 per gram. We are now maybe at 50, 60. And to get there, I think process intensification is very important. And especially on the downstream side, we have talked a lot about upstream, but on the downstream side, I think there is much more to do looking into unit operations that are not only chromatography, utilizing other technologies. And then it's really rolling out the short-term strategies for PAT, automation and digitalization, further acceleration and streamlining the CMC and regulatory part. And I'm not convinced that continuous is the answer for all questions. It's really based on the scenario, on the business case that will guide companies.
This has been fantastic, Andreas. With all that we've covered today, what is the number one takeaway you want us to walk away with?
Since we have talked so much about process intensification, I really want to underline that for me, the most important factor to drive productivity and to drive down costs, again, to leverage the full potential, it's important to understand the bottlenecks and choose the right technology. And there's not one solution that fits every situation. So it's really important to understand which position you are in to make the right choices.
Fantastic. Where can people get a hold of you, Andreas?
LinkedIn is a simple way to connect. Cytiva has also a landing page for process intensification with e-learnings, learnings about perfusion, upstream and downstream intensification, buffer management. And there's also an online tool that we call the Process Intensifier, where you can very easily simulate different scenarios in a process intensification journey.
Excellent. Smart Biotech Scientists, I will leave the links down in the description. So please reach out to Andreas and use these tools. And Andreas, thank you so much for being on the podcast. It has been a huge pleasure talking to you today.
Thank you so much for having me. It was a great pleasure. Thanks.
What a great journey through the future of bioprocessing with Andreas Castan. From continuous manufacturing roadmaps to sustainability metrics, we have covered the strategic insights that will shape our industry. If you have any complex bioprocessing challenges or questions you're wrestling with, I am here to help. Book your free consultation at BrühlmannConsulting.com/call. I am here to help you get started. No sales pitch, no strings attached, just help. So book your call. You find everything in the description. And please don't forget to leave us a review on Apple Podcasts or your platform you found us on. It will help us reach more people. Thank you so much. And I love hearing from you. I love hearing what you think about this podcast. And also, if you have other topics, also put that into your review. Thank you so much for tuning in today. Until next time, keep making bioprocessing smart.
All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.